Episode 6, First Spark, Origin of Life. What is life? This seems like a simple question, one that any child could answer. A rock is not alive. A tree is alive. A dog is alive. A cloud is not alive. We make these distinctions instinctively without conscious thought. But when we try to define life precisely, to draw a clear line between living and non-living things, the question becomes surprisingly difficult. Is a virus alive? It has genes, it reproduces, it evolves, but it cannot reproduce on its own. It needs to hijack. But it cannot reproduce on its own. It needs to hijack the cellular machinery of another organism. Is a fire alive? It consumes fuel. It grows. It reproduces by spreading. It responds to its environment. But we do not consider it living. Why not? Scientists have proposed many definitions of life, and none is perfect. But most agree on a set of characteristics that living things share. Life requires metabolism, the ability to extract energy from the environment and use it to maintain organization. Life requires reproduction, the ability to make copies of itself. Life requires heredity, the ability to pass information from one generation to the next. And life requires evolution, the ability to change over time in response to environmental pressures. Put more simply, life is chemistry that can copy itself and that changes over time. This definition might seem reductionist, even disappointing. Where is the spark, the vital force, the animating principle that makes life special? But there is no vital force, no magical ingredient. Life isn't magic, it is chemistry, complex and beautiful, but ultimately comprehensible chemistry following the laws of physics. And if life is chemistry, then the origin of life is a problem of chemistry. How do simple molecules obeying the laws of physics and chemistry organize themselves into something that can reproduce and evolve? How does non-life become life? This is one of the greatest unsolved mysteries in science. We do not know with certainty how life originated on Earth. We were not there to observe it. It happened billions of years ago, and it left little direct evidence. The first life was probably simple, perhaps just molecules in solution, with no hard parts to fossilize, no structures visible even under the most powerful microscopes. But while we don't know exactly how life began, we have learned that millions of years of evolution to every living thing that has ever existed. When we left Earth at the end of the last episode, the late heavy bombardment had just ended. About 3.8 billion years ago, the planet was scarred but stable. The ocean was permanent, no longer at risk of being boiled away by large impacts. The crust was solid, the atmosphere was thick with carbon dioxide and nitrogen, with no free oxygen, and the surface was bathed in ultraviolet radiation from the young sun, unfiltered by any ozone layer. This was not a hospitable world by any modern standards, but it was a world where chemistry could proceed without being constantly interrupted by catastrophe, and that made all the difference. Where did the life begin? This is one of the first questions we must address. The ocean covered most of the Earth's surface, but the ocean is a big place, and different parts of it would have had very different conditions. Some environments would have been more conducive to the origin of life than others. For many years, scientists assumed that life began at the surface of the ocean, or in shallow pools on the edges of primitive land masses. This idea, sometimes called the primordial soup theory, imagines the early ocean as a dilute solution of organic molecules, amino acids, and sugars and nucleotides, synthesized by lightning and ultraviolet radiation acting on the simple gases in the atmosphere. The experiments of Stanley Miller and Harold Urey in the 1950s seem to support this idea. They created a simulation of the early Earth's atmosphere, a mixture of methane, ammonia, hydrogen, and water vapor, and subjected it to the electrical sparks to simulate lightning. After a few days, they found that the mixture had produced amino acids, the building blocks of proteins, along with other organic molecules. This was a groundbreaking result. It showed that organic molecules, the stuff of life, could form spontaneously from simple gases under plausible early Earth conditions. It suggested that the ocean might have been a rich soup of organic compounds waiting for the right conditions to assemble into life. But there are problems with the primordial soup theory. For one thing, we know that the early Earth's atmosphere was probably not as rich in hydrogen as Miller and Urey assumed. It was more likely dominated by carbon dioxide and nitrogen, with little methane or ammonia, and in such atmosphere, the synthesis of organic molecules is much less efficient. More fundamentally, even if the ocean was full of organic molecules, it is not clear how those molecules would have organized themselves into life. The ocean is a very dilute environment. Molecules are spread out, moving randomly. They collide occasionally, but the most collisions do not lead to anything interesting. For complex chemistry to occur, for molecules to link together into chains and structures, they need to be concentrated, brought together in a small space where they can interact repeatedly. This is where the idea of hydrothermal vents come in. The hydrothermal vents are cracks in the seafloor where hot water from the Earth's interior is heated by hydrothermal vents are cracks in the seafloor where hot water from the Earth's interior is heated by contact with magma or hot rock erupts into the cold ocean. These vents exist today, and they almost certainly existed in the early ocean as well. At a hydrothermal vent, you have a steep gradient in temperature, chemistry, and energy. Hot mineral-rich water from below meets cold, relatively pure seawater from above. The minerals precipitate out of the solution, forming chimney-like structures around the vent, and within and around these structures there are pores and channels where water flows, where molecules are concentrated, where chemistry can occur. One particular type of hydrothermal vent, called an alkaline vent, is especially interesting for the origin of life. Unlike the hot acidic vents found at mid-ocean ridges today, alkaline vents produce warm alkaline water, and they create structures with natural compartments, tiny chambers separated by thin mineral walls. These compartments are intriguing because they resemble, in a very crude way, cells. They have an inside and an outside. They concentrate molecules, and the mineral walls that separate them can act as catalysts, speeding up chemical reactions, and as gradients, creating differences in pH and electrical charge that can be harnessed to drive reactions. Some scientists, notably Nick Lane and others, have proposed that these alkaline vent structures might have been the birthplace of life. The pores in the mineral walls would have served as natural test tubes, concentrating organic molecules and providing the gradients needed to drive the chemistry of life. This is still a hypothesis, not a proven fact, but it is a compelling one, and it has the advantage of being testable. Scientists are building models of these vents in a laboratory, trying to recreate the chemistry that might have led to life. Wherever life originated, on the surface or in the depths, it had to solve several fundamental problems. The first problem is the problem of replication. For something to be alive, it needs to make copies of itself. But how do you make a copy of a molecule? Modern cells use DNA to store genetic information, and proteins to do the work of copying that DNA and building new cells. But DNA and proteins are very incredibly complex. DNA is useless without proteins to read and copy it. Proteins are useless without DNA to encode their structure. Which came first? This is the chicken and egg problem of the origin of life. And for many years it seemed intractable. But then the scientists discovered RNA ribonucleic acid is a molecule similar to DNA, but with more important differences. Like DNA, it can store genetic information in the sequence of its building blocks, its nucleotides, but unlike DNA, RNA can also act as catalyst, speeding up chemical reactions. theory led to the RNA world hypothesis, one of the most influential ideas in the origin of life itself. The hypothesis proposes that early life was based entirely on RNA, not on DNA and proteins. RNA molecules would have stored genetic information and also carried the chemical reactions necessary for replication and metabolism. DNA and proteins would have evolved later as more efficient molecules for storage and catalysis. The RNA world hypothesis solves the chicken and egg problem. You do not need both DNA and proteins to start. You only need RNA, which can do both jobs, albeit less efficiently than the specialized molecules that will evolve later. But the RNA world hypothesis has its own problems. RNA is difficult to synthesize from simple building blocks. It is fragile, easily broken down by heat and hydrolysis, chemical reaction with water, and it's not clear how it gets from random RNA molecules to self-replicating RNA molecules capable of evolution. Despite these challenges, the RNA world hypothesis remains the leading idea for the early stages of life's origin, and there is an experimental support for it. Scientists have been able to create self-replicating RNA molecules in the lab, molecules that can copy themselves, albeit imperfectly, without the help of proteins. These molecules are not alive, not by any definition, but they are a step in the right direction. They demonstrate that replication, one of the key properties of life, can arise from chemistry alone. The second problem is that early life had to solve the problem of metabolism. Replication requires energy. You cannot build complex molecules from simple ones without putting energy into the system. Where did the energy come from? In modern cells, energy comes from chemical reactions, often involving oxygen and sugar or other organic molecules, but there was no oxygen in the early Earth's atmosphere. And while there may have been organic molecules in the ocean, they were probably dilute and not easily accessible. One possibility is that the early life used chemical gradients, differences in concentration or pH or electrical charge between different parts of the environment. At hydrothermal vents, for example, there is a natural gradient in pH where between the alkaline vent water and the acidic ocean. These gradients represent the potential energy that can be harnessed to drive chemical reactions. Modern cells use similar gradients, pumping protons into membranes, pumping protons across membranes to create differences in charge and pH, and then using those gradients to synthesize ATP, the universal currency, the universal energy currency of life. Perhaps early life used natural gradients in the environment before it evolved the machinery to create its own gradients. Another possibility is that the early life used minerals as catalysts and energy sources. Iron and sulfur, abundant at hydrothermal vents, can participate in chemical reactions that release energy. use iron sulfur chemistry, which may be relic of the earliest form of metabolism. The third problem is the problem of compartmentalization. Modern cells are surrounded by membranes, barriers that separate the inside of a cell from the outside environment. These membranes are essential. They allow cells to maintain different chemical conditions inside than outside, to concentrate molecules, to control what comes in and what goes out. But membranes are complex structures made of lipids, fat-like molecules that modern cells synthesize through elaborate biochemical pathways. How could the early life have had membranes before it had the machinery to make them? One answer is that simple membranes can form spontaneously from certain organic molecules. Fatty acids, for example, can self-assemble into spherical structures called vesicles with a bilayer membrane similar to that of modern cells. These vesicles are not as sophisticated as the cell membranes, but they serve the basic function of creating a compartment inside separate from the outside. Experiments have shown that RNA can become encapsulated by these simple vesicles, creating primitive protocells, structures that have some of the properties of cells but are far simpler. These protocells can grow and they can divide, albeit crudely. When they become larger or when the environment changes. Again, those are not living cells, but they are a step towards life, a demonstration that the basic architecture of cells, compartments surrounded by membranes, can arise from relatively simple chemistry. Putting it all together, we can imagine a scenario for the origin of life that goes something like this. In the pores of the alkaline hydrothermal vent, or perhaps in a shallow tidal pool on the edge of a volcanic island, organic molecules accumulate, concentrated by evaporation or by mineral surfaces. Among these molecules are nucleotides, the building blocks of RNA, synthesized by reactions driven by UV light or by chemical gradients at the vent. Some of these nucleotides link together in the short chains of RNA. Most of these chains do nothing interesting, but occasionally by chance a chain forms that has the ability to catalyze a reaction, perhaps a linking of another nucleotides into another RNA chain. This is ribozyme, a RNA molecule with catalytic activity. If this ribozyme happens to catalyze the formation of copies of itself, even imperfectly, then it can replicate. And if it replicates, then it can evolve. Chains that replicate faster or more accurately will become more common. Chains that replicate slowly or inaccurately will become less common. Natural selection has begun. Over the time, these self-replicating RNA molecules become more sophisticated. They evolve the ability to catalyze other reactions, not just their own replication, but also the synthesis of other useful molecules. They become enclosed in simple vesicles, membranes, made of fatty acids that concentrate the RNA and protect it from the environment. These vesicles containing self-replicating DNA and perhaps some simple metabolism are the first protocells. They are not yet true cells, but they are close. They can replicate, they can evolve. They are alive by any reasonable definition. From these protocells over millions of years, true cells would evolve. RNA would be supplemented and eventually replaced by DNA, a more stable molecule for storing genetic information. Proteins will evolve, more efficient catalysts than ribozymes. Membranes would become more sophisticated, with the embedded proteins that control the flow of molecules in and out. Metabolism would become more complex, harnessing a wider range of chemical reaction to extract energy from the environment. Eventually, these cells would diversify into the two great domains of prokaryotic life, bacteria and archaea. These are the simplest forms of life, single cells without a nucleus, but they are not primitive into the sense of being inferior. They are exquisitely adapted to their environments, capable of surviving conditions that would kill any complex organism, from boiling hot springs to the frozen soils of Antarctica to miles beneath the Earth's surface. And from these simple beginnings, all of the complexity and diversity of life on Earth would eventually arise. Every plant, every animal, every fungus, every protist, every complex organism traces its lineage to these first cells in the ancient ocean. When did it happen? When did chemistry become biology? We do not know precisely. The rock record from the earliest part of the Earth's history is almost non-existent, destroyed by plate tectonics and erosion. But we have some clues. The oldest rocks on Earth from a region in Canada called the Akasta gneiss date to about 4 billion years ago. They do not contain obvious fossils, but their chemistry suggests that they formed in the presence of liquid water, which means the Earth had a stable ocean by this time. Slightly younger rocks from about 3.8 billion years ago show hints of biological activity. The isotopic composition of carbon in these rocks suggests that some of the carbon may have been processed by living organisms, though this is controversial and not universally accepted. By 3.5 billion years ago, there is stronger evidence. Rocks from Western Australia contain structures called stromatolites, layered mounds created by mats of photosynthetic bacteria. If these structures are indeed biological, and most scientists believe they are, then life was not only present but already quite sophisticated by 3.5 billion years ago. This means that the life must have originated somewhere between 4 billion and 3.5 billion years ago, during a period of 500 million years or less, considering that the Earth is 4.6 billion years old. That means life appeared relatively quickly within the first billion years of Earth's history, perhaps even within the first 100 million years after the late heavy bombardment ended. This is significant. It suggests that the origin of life is not an incredible, unlikely event requiring billions of years. extraordinary circumstances. Rather, it seems to be something that happens relatively easily given the right conditions. Water, organic molecules, energy, and time. Put them together and life emerges. This raises a profound question. If life arose so quickly on Earth, might it have arisen somewhere else? Are we alone in this universe, or is life common wherever conditions permit? We do not know the answer. We have only one example of life, the life on Earth, and all of it shares a common ancestor. We cannot say whether life is common or rare in the universe based on the sample size of one. But the fact that life arose so quickly on Earth is encouraging for those who search for life elsewhere. It suggests that if we find planets with liquid water and the right chemistry, we might find life. Mars may once have had life in the ancient past, where it had liquid water. On its surface, Europa, a moon of Jupiter, and Enceladus, a moon of Saturn, both have subsurface oceans that might harbor life, and the thousands of exoplanets we have discovered around other stars. Some of them are in the habitable zone where liquid water could exist, might also have life, but this is speculation. For now, we only have Earth, and the life on Earth, despite its incredible diversity, all shares certain fundamental features that point to a common origin. All life uses DNA or RNA to store genetic information. All life uses the same genetic code, the same mapping between DNA sequences and amino acids. All life uses ATP as the energy currency. All life is based on cells, but these universes suggest that all life on Earth descended from a single common ancestor, an organism that lived billions of years ago, perhaps not long after the origin of life itself. We call that organism Luca, the last universal common ancestor. Luca was not the first living thing. It was a survivor, the organism whose descendants outcompeted all of the others, whose lineage persists to this day in every living cell on the planet. Before Luca, there may have been other lineages, other experiments in life, but they all went extinct, leaving no descendants. We can infer some things about Luca by looking at the features that all modern organisms share. Luca probably used DNA and RNA. It probably had a cell membrane. It probably had ribosomes. The molecular machines that build proteins. It probably had some form of metabolism, extracting energy from the environment through chemical reactions. But Luca was also probably quite simple compared to modern organisms. It may have had fewer than 1,000 genes compared to the tens of thousands in a human cell. It probably lived in a warm, chemical-rich environment, perhaps near a hydrothermal vent, and it was almost certainly single-celled. From Luca, life lineages evolved different strategies for survival, different metabolisms, different structures. Some became the ancestors of bacteria, others became ancestors of archaea. Much later, some archaea would merge into bacteria in a symbiotic relationship that would give rise to a third domain of life, eukaryotes, the complex cells that include all plants, animals, fungi, and many single-cell organisms. But that story is for a later episode. For now, in this episode, we celebrate the origin of life itself, the transition from chemistry to biology, from non-living molecules to self-replicating, evolving organisms. It is one of the most important events in Earth's history, the event that transformed our planet from a geological world into a biological one. And it happened quietly, without fanfare, in some forgotten corner of the ocean, of the ancient ocean. No one was there to witness it. No one knew that something extraordinary was happening, that a threshold was being crossed. But it happened. And once it did, the Earth would never be the same. The story of our planet would no longer be just a story of geology and chemistry, of volcanism and erosion, and the slow drift of continents. It would become a story of life, of evolution, of the endless creativity, of natural selection playing out across billions of years and billions of species. In the next episode, we will see what the first life was like, these bacteria and archaea that dominated Earth for the next 2.5 billion years. We will see how they survived, how they evolved, how they began to change the very chemistry of the planet. We will meet the invisible empire, the microbial world that built the foundation for all life to come. But for now, we pause here at the moment of life's origin, in awe of the fact that it happens at all, that chemistry can organize itself into biology, that non-life can become life. We are the descendants of that first spark, that first self-replicating molecule in the ancient sea. Every cell in your body carries echoes of Luca, the same basic machinery, the same genetic code. You are connected through an unbroken chain of reproduction, stretching